Here we go, I think. Okay. Okay, there we go.
I hope they don't see that.
Avery, do you want to go over this since I'm about to talk for a while?
Okay, so the first thing we're going to do is obviously with the university's emails and everything being super up in the air right now, we are getting a little bit more solid confirmation for what rules are, what things we're allowed to do. We're going to give you the update on our plans for the semester. Us as an exec team have made some decisions to what we feels best to keep everyone safe, but give you guys stuff to do this semester. And then the other thing that we're going to do is some fun stuff. We're going to do Scientist Spotlight today, where Hope's going to tell you about some super cool stuff. And then we're going to do research opportunities, where some of the exec team that's in labs are going to tell you about what we do. You guys are welcome to ask us questions about like the process of getting into labs, what we like about it, what we don't, stuff like that. And then it's going to be some wrap-up surveys and questions, and that's going to be it for the day. So that is the agenda. Let's go. So the not good news, depending on what you guys think, is we have decided to not do anything in person this semester. That is both obviously for everyone's health, safety-wise, and some of us just don't have strong faith in the university staying in person throughout the whole semester, based on other decisions that universities are making. So we're definitely going to be all online all semester. That's the most accessible option for everyone, and it's just the most solid plan that we can make right now. Our in-person events with the Hefner Museum have also been postponed for a later semester, since obviously that requires a pretty strong commitment to be on campus, to attend trainings, things like that, and they're struggling with their own health safety guidelines and things like that.
Happy.
The good updates is that we are definitely going to have guest speakers. We're coordinating that right now. We're going to do our grad student panel, so you guys are going to get to ask them about stuff, and that was an awesome experience last year. We hope you guys are going to love it this year. And we're still going to have these online meetings so that you can make friends. We're going to chat, and it's just generally going to be a fun time. So let's go on to Scientist Spotlight. And this is Hope's.
Wait, can they see me? I don't want to.
I believe they can see you.
Yeah.
Sorry, I don't want to awkwardly be on like half, and you can see like half my face the whole time. That'd be a little awkward. So today I wanted to talk about not necessarily a single scientist, because there were a lot of scientists involved in this, but overall just talk about genetic cloning. So I'm guessing not everyone knows what genetic cloning is, which is 100% fine, which is why we're going to go over it. So first of all, can someone, do you guys like know what plasmids are? Did you guys learn what plasmids are yet?
I know what they are from AP Bio in high school.
Can you tell us what they are then? Perfect.
They're, I think if I remember correctly, they're like sections of DNA in bacteria that usually they code for like antibiotic resistance and other things. They can be transferred between bacteria.
Beautiful. Thank you so much, Alex. Yeah, so plasmids are basically just an extra chromosomal version of DNA that usually holds like non-essential genes. And the unique thing about plasmids is that, like Alex mentioned, they can be given to different bacteria, whether among bacteria, or what we can do in a laboratory setting is basically take a plasmid and force bacteria to take up the plasmid for themselves. There we go. So in other words, plasmids provide a really unique method of introducing DNA, such as a gene that we might be interested in, into bacteria and having them do the replication for us. So as you might imagine, scientists soon took advantage of the opportunities that plasmids provided. So what they were able to do is they were able to edit these plasmids that they had using something called restriction enzymes, which were basically like scissors that cut very specific sequences of the DNA. They would cut them at those points, and then they could stick these fragments that they made back together using ligation reactions, so using ligase from a certain phage or bacteriophage. And so they were able to basically cut it up and then put them back together in whatever order they wanted, as long as they had the proper restriction enzymes and they did it in a certain way. So again, in other words, basically what that allowed is plasmids would provide a method of us as scientists introducing DNA into bacteria and having them replicate whatever DNA we wanted them to. So this basic idea seems relatively simple, because it is relatively simple. And ironically, was actually thought out by a couple of scientists who met up in Hawaii for a conference for plasmids in general. And literally they were just getting dinner and were discussing stuff. And they were like, wait a second, we could do this. And that's literally what they did. Like literally just a week later, they were like, yeah, let's try it. And sure enough, it actually worked. So after that point, it was basically the idea of, okay, this system generally works where we can form a plasmid and basically put it into bacteria, but we want to do this as efficiently as possible. And so that's where some other people came in, and specifically that's how we came up with the plasmid PBr322. So that stands for P, plasmid, BR is standing for Bolivar and Rodriguez, and then 322 is just a random beautiful number that they chose. But PBr322 is the plasma we're going to talk about. And so the main goal that they had with this was specifically they wanted to create this cloning vector, which is just a fancy word for basically saying a plasmid that you can stick whatever you want in, with one restriction enzyme site, which I'll get into why that's so important, and then with a specific ampicillin resistant gene, and then 4 unique restriction sites. So the reason this was so important, we'll get into later. Sorry, I thought I had another slide. Nope, I didn't. Okay. So the way they did this is basically how I talked like in the beginning with the scissors and the glue, where they basically just did it multiple times, where they took the original plasmid of the time that we use most often, PBr313, and they just took it, put restriction enzymes with it, and then ligated it again. And they basically came up with a bunch of plasmids, and they randomly found PBr322. So PBr322 was specifically unique for several reasons, and we'll get into it starting now, but this basically became the crux of genetic cloning. It provided so many opportunities, and there's so many different things we've gotten from this one plasmid. So one thing that it has a single PST1 site. So PST1 is a restriction enzyme, so it's like the scissors. So that meant that if you put PST1 into a mixture, it would only cut at this one site. So this allows for really easy detection, because what you can do is you can cut at that site, put in a gene that you want, and all of a sudden something that originally was resistant to ampicillin is no longer resistant, because the thing that they had can no longer be transcribed because there's a big gene in the middle of it. The second thing is it also provided easy recovery. So the fact that you could have it right there also means that you could very easily cut it back out and retrieve it, which was really nice for cloning, where basically we just wanted them to replicate a bunch of that gene and then just take it back and get it for ourselves. So the other thing is bacteria, again, Alex actually hinted towards this earlier, bacteria are really good at sharing. So what they'll do is a lot of times they'll go through conjugation, if you've heard of that, where they'll basically share their DNA, specifically plasmids, with one another. Yes. The other thing that they can do is do homologous recombination, which is basically they can take a part of the plasmid and integrate it into their chromosomal DNA. Now, this causes problems for cloning because we only want certain bacteria to have it. We want to be able to tell that only those bacteria have it. And so if it's able to integrate it into its own DNA to the point where they no longer have the plasmid and they can get somehow resistance, or they can just share it with one another, that's failing our system, and we don't like that. So the unique thing with PBr322 is they basically had non-transposable elements, meaning there was no homologous recombination. If they're in the plasmid, they are staying in the plasmid, and there was less mobilization and conjugation taking place, so there was less sharing, just naturally, compared to all the other plasmids that they had. No. Okay. So the other thing is basically at this point, we had only had a couple of antibiotic resistant genes that we had put on this. And for every antibiotic resistant gene, that's basically another way to detect and another place where you can add a gene. So it basically, you want more resistance. That way you can have more variety and do a lot more things. So up to this point, they didn't really have a lot of variety. And then PBR 322 came along and suddenly they had a lot more options. And then from PBR 322, they would make even more, like PBR 325, which is a lot more commonly used now, where basically it had a bunch of resistance probabilities, a lot of specific restriction sites. So you could do a lot with it. So the final thing that's really unique about PBR 322 is, as you can see, it's physically tiny. It is really, really small. And that's awesome because that means that it not only has an advantage for us as scientists, because we can just put a bunch of things in and not worry about it becoming humongous, but it's also nice for the bacteria because it's a lot easier to copy. The other thing is it has a high copy number, meaning rather than just copying itself a couple of times, there's a lot of it, which is nice because we want the genes to be copied. So now we have a bunch of the gene. So it's small but powerful. So ultimately, PBR 322 has become like a crux in molecular cloning and has really become really important in any kind of genetic setting that you would witness, whether directly or from certain derivatives that we've had from it, such as PBR 325, to the point where most of the plasmids used in a genetic lab actually somewhat originated from PBR 322. So once again, the really unique thing about this entire thing was the fact that this was all accidental. People thought of this by just sitting in a room, thinking in Hawaii of all places, and then they just took advantage of it and they started actually moving forward in this. And now we have entire systems like medications, insulin. If you have diabetes, you are surviving based off of this concept. GMOs, we feed almost entire countries based off of this technology. I already said that. We can use bioremediation, which is basically oil-eating bacteria, clean up our environment with bacteria by this technology as well. Rice, again, feeding a bunch of people who need help by genetically modifying certain foods. And this is literally something that you can buy online. Like, plasmid kits have become just an essential factor in most genetic labs. So it's really unique that this one little thing that seemed to be an accident has basically developed an entire system that we base genetics on right now. So it's really, really cool. But yeah, so even if you don't remember the names, which is fine, just remember that plasmids are amazing, bacteria are amazing, and yeah, and so are you. So sorry, I had to. That was perfect.
Oh, you're so cool.
Yeah. Does anyone have any questions? Great. Well, thank you, Ho. That was very wonderful. Now we'll go on to like different research opportunities in the microbiome department. A good chunk of us in the exec group is now in a research lab, so we know the ins and outs. This is also to help you like kind of figure out what's out there, which is always nice to know, like your different options. You can always go online too, but it is nice to kind of hear about it and just kind of figure out what you like, what you want to get into. Obviously not right now, but okay. Ariel is not here because because physics is fun. But she actually is not microbiome, she is bio, and she does work with regeneration specifically in newts. So she gets to touch little newts for fun. So that's awesome. I'm sorry, I don't know much about hers. I know it's cool and she does a lot of neat stuff with these little guys, and look how cute they are. How could you resist? How could you resist? Haley is also not here because physics. She actually does not even do research in a STEM field. I believe she's doing comparative religion studies right now.
I can explain a little bit about hers. If you want.
Yeah, please.
So Haley's is obviously more on the behavioral side, and the specific project is Preaching Goes Viral. And so it's a research study that's meant to evaluate how different religions are interpreting and reacting to the COVID-19 pandemic, and also how different people of those religions are being treated during the pandemic. So I know a rather recent one is, I'm not sure if I can see it on the slide because there's a thing like right where I think it is. But for instance, there's issues that already a lot of Islamic healthcare workers face discrimination in the workplace, and COVID-19 has really exacerbated that, along with the fact that a lot of sects of it demand, it's a very, like how their religion, like they pray multiple times a day, there's very strict demands on their time. With COVID being in full like PPE suits, you can't just step out of the room and go do that. So there's a lot of societal questions going on right now of how to deal with religious demands versus healthcare demands, prioritizing like safety over like belief, and how to make compromises between those two things. Do you exclude people from the workplace? Like trying to find solutions to that, basically. So that's what she works with, and it's interesting. If she's here another time, I recommend asking about it. So it's cool.
It's also one reason that we wanted to bring this up too is the fact that just because we're like a STEM major or something does not mean that you need to do research in STEM fields. Obviously, that's preferred in the sense of if you're interested in doing this for the rest of your life, then you probably want to see what it's like by looking in lab. But at the same time, you can very easily, especially now, you probably are much more likely to be able to get some kind of research quality thing out of your maybe non-STEM situations, such as Haley's able to do now. Like everything she's doing, she can do online pretty well. Whereas like for microbiome stuff, it's a little harder. It's a little bit more difficult to come up with stuff. So do not in any way feel like, oh, it needs to be this exact thing if I'm going to get into school. It's like, no, research has a general idea, a general mindset that is the same along any kind of field. And so if you're interested in a topic, do not let common courtesy or rules that seem to be there dissuade you from doing that. Also, Haley, again, is not here because she's also taking the physics exam, but she wanted to say that if you have a response, a religious response, anyone can post anything on the website. So if you want, you can feel free to email her, and I will put her email in the group chat real quick. But yeah.
Okay, so this is mine, and on the topic of exploring basically as many different departments as you want to, I have done research projects in 3 different departments at our university in my 3 years here. So the photos on the right side are my previous research projects. The top is the Blue Jay photos. This was a project that I actually collaborated with Maddie on, and I like collaborated as in we both literally did the entire thing together along with another student. So that was in the biology department under Dr. Russell. It was a very brief kind of 1-year project where we looked at blue jay characteristics to try and determine if there was physical differences between male and female versus a hatchling and an adult. So like those 4 kind of categories. Because looking at those birds, the bigger one on the left is not the adult male. Like, so it's very difficult to discern between them. It's very important for population studies on those birds to try and tell what kind of bird you're looking at. So that project was a lot of kind of data mining, organizing. I think it was done entirely on a laptop, so I unfortunately did not get to hold birds. But that was an interesting project. The bottom right photos is a peony flower, and this project was in the botany department, also under Dr. Russell. Basically, what I evaluated is mycorrhizal fungi, which is the picture on the right. They basically do symbiosis with roots of most plants, but it's unclear if they do it with peonies. Objectively, what we know about them says they should, but the structure of peony roots are really, really difficult to analyze when it comes to microscopy because they're bright pink. So you can't stain and dye them. You can't really see structures in them. And they also fall apart like tissue paper when you try to handle them. So my project with that was just trying to determine if mycorrhizae fungi were there, and if they are, what commercial value might they have to peony farmers. Then my actual long-term, like related to my major project, is with Dr. Bowman, and those are the photos on the left side. Maddie also works in this lab, and basically what our lab focuses on is ammonia oxidation, metabolic pathways. So there's certain bacteria and some archaea that, rather than do like sugar digestion, like what you might think of, they do ammonia oxidation for their metabolism. They detoxify ammonia into nitrate. And this has a lot of different like purposes that can be used for like water treatment mostly. It is a major factor in like the global recycling of nitrogen, which is how we support life on this planet. And the interesting thing is that they were considered completely unculturable in lab until about 10 years ago. So they're a very novel area of microbiology to be working in. The specific project that I do is metabolic research on them. So my project last year was plating them with different amounts of sugar and proteins and different things like that, just to try and see if they can even uptake those kinds of things like from their environment. Because obviously their metabolism is super insane. And so people don't really know what they're capable of. Since the recent shift online, me and Maddie are again collaborating on a project for this semester. And our most recent one is we do genetics work. So we're using a bunch of online analysis programs we basically are trying to figure out a lot of different stuff about the genetics of our bacteria. Since they're so new, it's unknown like what proteins they have, what all different like metabolisms can they conduct, what genes do they have that are their own versus they might have gotten from another species through conjugation or horizontal transfer, things like that. So yeah, I have done many projects over the years. I'm happy to talk about bouncing in between them and things like that. And that's what I'm currently doing.
Just going off of what she was talking about with the genetic stuff, I think it's really cool in that we're not looking at just genomes like by itself. Like we just pick out like an ammonia oxidizer and look at it. We actually take the genome, the metagenome of everything that's in the culture. And basically what we're doing now is like picking out what is what and just figuring out what is hanging out with these ammonia oxidizers and why, which I think is really neat. When I am in lab, I study stress factors on these ammonia oxidizers. So specifically hydrogen peroxide. Hydrogen peroxide, if you don't know, is a radical oxygen species, which basically means it's not a good time. It's going to damage your DNA and you're just not, it's not fun. So I basically get to poison the cells that I work with, see how much hydrogen peroxide that they can handle before they're like, kill me. Yeah. Oh, that's a morbid way to describe it. But yeah, it is a lot of fun, and maybe I'm just a sadist, who knows?
Our lab is fun. Our training involves how to not accidentally make cyanide out of the stuff we throw away. Our lab is exciting like that.
All right. I think, we're running out of time, so we're going to go ahead and jump to Hope, if I can make it go. There we go.
So I work in a primarily genetics lab with Dr. Actis. And so basically what we look at is the pathogen Acinetobacter baumannii, and it's a pathogen that's very commonly infecting immunocompromised people. It's not a good bug. But what we particularly look at is specifically BLSA, which is a certain photoreceptor that they found in this pathogen So basically, this pathogen has a lot of things that are regulated by light, which is insane. Like, just what? Like, why is a pathogen using light as a source for regulation? And so basically what we're doing right now is we're trying to further characterize BLSA as a photoreceptor and see what else it controls, both at 24 degrees and at 37 degrees. So both when it's hypothetically inside the body and out. Why there's light regulation when it's inside the body, we don't know, which is part of the reason why it's so insane and cool. So basically just trying to further characterize those systems through both biological tests as well as looking at the genetics and trying to understand it at both a biochemical, protein, as well as genetic level.
Neat. Do we want to do one?
So one thing that we wanted to do is, obviously research is a thing of confusion right now, and so we talked about this briefly the very first meeting we had, but we wanted to also just inquire again, would you guys be interested in basically us doing as a club a project where we would basically make a poster? So you would be able to put on your resume and post on your LinkedIn, your Handshake, whatever, this is mine, I made it.
If you guys could put like a yes or no in the chat, we'd really appreciate it.
We're not gonna, it puts a little bit of effort on our part, but ultimately we would all work on it together. Okay,
That sounds awesome.
common trend here.
Cool.
Okay, perfect. We'll make sure that that's in the works.
Yes, that is exactly the idea. Helpful for grad school and if you need a resume for lab or anything like that.
Mm-hmm.
Awesome. Do we want to ask anything else? I think that's all we wanted to ask about, I believe. Oh my gosh, I can't do that. Okay. Does anyone have any questions about anything ever? Life? No? Okay. All right.
Show you
Well,
pets
thank you
again.
guys. And here is the date for the next meeting. Put it in your calendar. Come meet us, please. We want friends.
Please.
Probably it's dark. Oh no, there we are. Neat. Does anyone have any exams for tomorrow? What?
Are we gonna get cut off in 3 minutes?
Yes,
3 minutes and 30 seconds.
still working on it.
The
Okay.
goal is that this won't be a problem, but for now
Well-
we're going with the free account.
Have we ever waited to see what happens?
I have in another situation,
You did?
and it
Okay.
did not end well. You're just
We're just
everyone
gonna
dies.
absolutely. Everyone's dead. Okay, awesome. Does anyone have anything they want to talk about in our remaining 3 minutes on this earth?
Uh, uh,
No pressure.
I have this.
You have
two-headed
what?
monkey.
I don't know what that is.
Oh, it's a monkey.
It's a two-headed monkey coconut.
Cat. There's a cat. She's sleeping now.
One time I was watching this TV show on the like Science Channel or something, and they called the Black Plague a virus, even though like it's a bacteria.
Nice
Everything
cat.
on the Science Channel is like cryptids now and ghosts.
They got disavowed because of that incident.
Love
Speaking
science.
of ghosts, I was watching Ghost
Reputable
Adventures
sources
today.
can be wrong on very basic things.
Very scary. Very scary.
I, for years, said that influenza was a bacteria because I saw it reported wrong on the news one time.
Yeah, like I read a book about the 1918 influenza, and they actually thought it was a bacteria because whenever they checked the lungs of influenza patients that died, they had bacteria in the lungs.
Ooh.
I thought you meant that the person who wrote the book thought they were bacteria.
No,
Like,
people
oh,
in
no.
1918, the scientists, they thought it was a bacteria.
I'm pretty
easier
sure
in
it's a
this world.
bacteria, guys. Oh, that's crazy.
That's awesome that you're interested in those kind of books, though. Sometimes
For
it
sure.
takes a while for me
Yeah. Yeah.
Sometimes I get all scienced out from all my courses. And I'm like, I don't want to learn.
Sometimes it takes a while for me to read a book.
Yeah.
I was good over quarantine. I read some books over quarantine, but
I'm proud. Yeah.
I still have to read Lord of the Rings, Hope.
Oh,
I don't
God,
know if I
that's in a
told
minute.
you. I think I messaged you that I got it, but I
We have less than a minute left.
Will it keep my recording if it ends us? Okay.
I don't know.
Think. Oh, that was scary. That would be bad.
All right, bye everyone.
Bye.
Unless we get
Awkward.
upgraded.
Bye.
Bye.
Bye. Who knows how long we have? I want to stay on and see what
I want to
happens.
see. I want to see what
Yeah,
happens.
same.
Is it really just gonna lead us out, do
That's
you
funny.
think? Like a big
Well, we're gonna test it. I wish there was a countdown so we could be like 10, 9, 8.
It's gotta be near in a minute.
Right? Yeah.
This is like 10 seconds. Like, what? Start, come on.
Come on, tell us.